Hi class, in this video we're going to focus on your exercise 25 content, which are the structures of hearing and equilibrium. We have our ear broken up into external, middle, and inner portions. And within the inner portions we have the cochlea, which is used for hearing, and the vestibule and semicircular canals, which are used for our sense of equilibrium. Sometimes it's referred to as the vestibular system. And we can see these structures pretty nicely in Visible Bodies Anatomy Atlas. So I'm just in the microanatomy view, and I'm just going to go ahead and click on ear. So let's dive into it. While we're looking at our lab objective sheet, we have the external ear to start off with. Our external ear starts with the auricle. So I'll go ahead and just highlight the entire auricle. Another name for this that you'll see in the literature is pinna, and we're just going to focus on the entire auricle as a structure. We're not asking you to know the substructures of the auricle. And then from the auricle, sound waves will travel here through this passageway, referred to as the external acoustic meatus, sometimes also referred to as the external auditory meatus. You know, when I said we weren't going to ask you to know substructures, I misspoke. There was one substructure of the auricle we do want you to know, and that's the lobe. The lobe is the part that dangles down right here. That lobule is the part that hangs down. Sometimes it merges with the body, the side of the neck. Sometimes it will be extending outward. And that whether or not the lobes of your ears are attached or detached is one of those classic traits you look at for dominant recessive phenotyping when you're learning Mendelian genetics. Anyways, moving on here, we have the external acoustic meatus, and that external acoustic meatus goes to the tympanic membrane on the innermost portion of the ear, right there. This tympanic membrane, sometimes, or in common English referred to as the eardrum, marks the dividing line between the external ear and the middle ear. On our lab objective sheet, we included the tympanic membrane with the middle ear. Every once in a while, depending on how your lab manual categorizes it, the tympanic membrane will be included with the external ear. I think of it as a dividing membrane. The root word here is tympan, and it means drum. For those of you who have any kind of orchestra or band experience, you may have heard of the tympanies, which are large brass or large copper drums. They have a very distinctive sound. Connected to the tympanic membrane, we'll have the auditory ossicles, or ear bones of the middle ear. So now we're looking at the middle ear. And the first bone is the malleus. The next, we'll go ahead and highlight the entire malleus right there for us. The next bone is the incus, and the final bone is the stapes. In lab, since this is a college course, we're going to stick with technical terminology, so don't use the hammer, anvil, and stirrup. We want malleus, incus, and stapes for these bones. And we're only asking you to know the whole bone. We're not asking for substructures of the bone. These three bones are going to carry the force of sound waves or vibrations that are picked up by the tympanic membrane, and they'll carry those sound waves all the way over to here, to a structure that's completely covered by the widest part of the stapes. That is the oval window. And at that point, we transition to the internal ear. But before we dive into the internal ear, there's one more structure in the middle ear that we need to cover, and that's the pharyngotympanic tube, formerly known as the Eustachian Tube. So while we're here in the middle ear, let's go ahead and, oh, come on, Visible Body, you can work for me. There we go. We'll zoom out. We have the Eustachian Tube. And I'll admit that's how I learned it back in the day. There are some other names floating around for this tubular structure, one of which is the pharyngotympanic tube. That's a pretty good name because we have if I were to zoom out here, let's go ahead and clear it and give us some good reference points. Right about here, where I'm putting this circle, we would have the pharynx. And up here is the tympanic membrane. So to call it the pharyngotympanic tube is to say that the pharynx is connected to the tympanic membrane by this passageway. It's worth emphasizing that in adults, the tympanic tube has a good incline to it, so it's a nice steep angle. But in young children, the Eustachian tube tends to be more horizontally oriented. And because it's more horizontally oriented, it doesn't allow for good drainage from the middle ear. Consequently, young children are more likely to suffer from ear infections, or what are referred to as otis media. Anyways, let's move on to the internal ear. The internal ear is broken up into a cochlea, a vestibule, and semicircular canals. So the three main structures. And we can highlight those structures here. They are shown in kind of a bluish gray or grayish blue. We have the vestibule in the middle. We have the cochlea as the spiraling seashell-shaped structure. And then we have these three tubes or passageways that make up the semicircular canals. And I want to emphasize with these semicircular canals that they are all perpendicular to each other in three-dimensional space. That's important because it's going to help us with our sense of dynamic equilibrium. And because we have these three tubes that are perpendicular to each other in three-dimensional space, fluid can flow through them to different degrees to help us regulate and have a sense of our dynamic equilibrium. So let's look at the cochlea in more detail. The cochlea is right here. Connected to the cochlea, we have the cochlear nerve, and inside of the cochlea, we have the spiral organ, formerly known as the spiral organ of Corti. And if I slip up and start saying spiral organ of Corti, it's because some of these newer terms in our lab manual I'm not as comfortable with. They haven't been embedded in my brain like the old school terms. So here I'm pulling up a cross-sectional view of the cochlea, and we can see here the cochlear nerve. That cochlear nerve is going to connect to some cells that have some hairs coming off of them right here. And as these hairs vibrate back and forth, we will take those vibrations and turn them into action potentials that we send down the cochlear nerve to ultimately be interpreted as sound. Now, as we're looking at this structure right here for the spiral organ, I want you to focus on this structure that I'm outlining. This is our spiral organ. This spiral organ is what's going to take the vibrations that are sent to the inner ear and convert them into action potentials that are interpreted by our brain as sound. On our lab objective sheet, let's pull that up here. We're not asking you for the parts of the spiral organ, so I'm not going to spend any more time talking about the spiral organ. You'll get that in the lecture portion of this course. Let's go back to the lab objective sheet and go to the vestibule. So we'll back up here and click on inner ear. Here's the vestibule. And when we look at the vestibule, I really wish that I would have the ability to hide structures here, but in microanatomy views we can't. But what we see here is that the stapes presses up against the vestibule, and when there's a vibration, as the stapes will push in or push out, it will push against a soft membrane that's directly beneath it. That soft membrane is called the oval window. So called because of the oval shape at this large edge of the stapes. So you may be asking yourself, well, how can you ask me about the oval window if you can't show me a picture of the oval window? And that's a pretty good question. On a lab exam, I would indicate this area either with an arrow or an oval, and I would ask you to identify the membrane beneath the bone. And the answer to that question is the oval window. When the oval window is depressed by the stapes and pushes in, it causes a slight increase in pressure within the vestibule, and to relieve that pressure, we have another window. This is referred to as the round window, which is right here and visible within visible body. So when the stapes is pushed in, the round window bulges out, and that will equalize pressure within the inner ear. I'll go ahead and click on the round window right now. Come on. There we go. There's the round window for us. Connected to the vestibule, we have the vestibular nerve. That vestibular nerve is going to carry our sense of equilibrium from the vestibule and from the 3 semicircular canals to the brain to allow us to have our sense of equilibrium. So here is the vestibular nerve right there for us. And then the vestibular nerve and the cochlear nerve will merge together to form the vestibulocochlear nerve, which is one of the cranial nerves that you need to know. That is everything for the ear on ear anatomy. If you have any questions or comments about ear anatomy, please feel free to post them in the class discussion board or to shoot me an email. And as always, happy studies.